about the way we produce those hormones, where those hormones come from, when they come from. So remember, this human chorionic gonadotropin comes from the embryo, hence why you can determine that there's a pregnancy, because it's coming from the embryo. And it's a big flush of the human chorionic gonadotropin, but see how it fades down? What is it that is developing right around 12, 16? What is it that's developing down in here that we don't need that information anymore?
The placenta?
The placenta, exactly. So the placenta now is coming on, and the placenta is taking over the job of the corpus luteum, because corpus luteum has to make all that progesterone and estrogen in the early days. But once that placenta is going, then we have our progesterone and estrogen that is being produced by the placenta. And it's this progesterone, remember, that's really important for maintaining that uterine lining and preventing any contractions, so we can maintain that pregnancy. Let's see, who else should have pictures here? Should I see what there she is? Belinda's here. Okay. So, we got that going. This is another way of looking at the same information. I kind of like it because it does tell you along the length of the line time frame, human chorionic gonadotropin, done after the 4th month of pregnancy. You already know you're pregnant, so that's cool. Then you got your corpus luteum, and that's taking place, giving us the progesterone and estrogens, maintaining our endometrium especially, and getting our mammary glands ready, because that baby's mother needs to be ready for the baby. Corpus luteum is going to then be taken over by the placenta. Relaxin, of course, really important that the pelvis and the pubic symphysis relaxes a little bit, because you got to have a little bit of give and take in that pubic symphysis while parturition's occurring. And we got the human chorionic somatomotropin. Say that one fast 10 times. Our glucose. Remember this discussion about how this has everything to do with insulin and insulin production, and the fact that this can lead to gestational diabetes. Corticotropin releasing hormone, that is going to be important for the cortisol, which is going to be important for the beginning of birth. There are some early pregnancy tests available out there. Maybe some of you are aware and have actually used one somewhere down the line. No, but some of you might have. They have good results. They're fairly easy to use. There can unfortunately be false negatives and false positives, and I was curious about those false negatives, false positives, and the false negatives and the false positives have the same reasons. And one of those is that you test too early. So remember, day 5 is when that human chorionic gonadotropin starts to come on, but maybe you need to wait a few more days to get a positive result. Diluting your urine may be another reason for having a dilute urine, using a expired test, maybe not covering the entire area that needs to be covered with urine. You might have a false negative, false positive on that regard. That would be improper use of the test. Chemical pregnancy. Certain medications, hormones, imbalances can all lead to false negatives, false positives. Things that other might not have considered if you have a lot of protein in your urine, or you're on steroids, or you're on diuretics, which would have the result of a dilute urine. I don't know what thyroid drugs would do to it, but all of those things might impact the results that you're looking for. And you remember in lab when we were talking about this development process? This is fascinating to me, that we share so many genes with so many creatures across the planet. The genes that we share are called conserved genes. We talked about how you can have that conserved gene for driving your appetite, right? Neuropeptide Y. And that drive to eat, of course, spans many different species, because we all got to get food to make energy. This is just showing how we share certain genes with the Drosophila. This is a fruit fly, and the Drosophila's genetic makeup and our makeup share these genes called Hox genes, or homeobox genes. Have you heard of them? Did anybody watch that little video, the evolution? No, it's really fascinating to see how the lay of the land, the lay of the body, is dictated by these very first genes, the first ones that get turned on. And they determine the position of the head, the position of the thorax, the position of the abdomen, the position of the tail, the position of the limbs for all of us. So you can do a color coding here, color mapping here, and see there's our thorax, there's our abdomen, there's our tail, there's our limbs, all based on genetics. Very cool. So you can learn more about this if you'd like to see more about Hox genes. And I was talking about this too. This is an old image, but it does kind of give you a sense about how we embryologically really look a whole lot like each other, like the fish, the salamander, the human, right? Isn't that amazing? And this is true stuff. But as we develop, of course, we start to differentiate. And that differentiation is, of course, based on genetics. Yeah, which genes get turned on when? What genes do we have? We share genes with chickens. We share genes with salamanders in terms of lay of the land. Banana. Bananas. Not so many bananas. We don't share genes. This is an image, as you can see, a scanning electron micrograph of the embryo of a monkey, and it's just interesting to note that we have the head region already detected, already determined, the tail region already determined, right? That's week 2. Head, tail, head, tail, head, tail, head, tail, week 2, already established. The somites that are going to establish the vertebra and some of the musculature. You have the neural plate, which ultimately will become the brain. You have the amniotic sac that's surrounding this whole structure, filled with amniotic fluid, neural folds. All of this is going to become your spinal cord and nerve, and spinal cord and vertebra that surround the spinal cord. Just another image of the same thing. And in terms of the primitive streak, maybe did you watch the first 3 weeks where you talked about developing the primitive streak in the notochord? Yeah, so here we have our neural groove, we have the primitive streak, and again, establishing the head and the tail region. Kind of interesting also to note that the heart starts pretty early on, like within the first 3 weeks, you start developing a heart. And that heart, of course, is not the full 4 carat full chambers, but rather a single chamber and a single chamber. But the key here is that you have those cardiomyocytes that are autogenic, that they contracting all on their own, those little cardiomyocytes. Yeah. Another image, what's this? Oh, about 1 month. The limbs are starting to show, the buds are starting to show. The brain and spinal cord are getting established. Optical vesicles, part of the eye is showing up. The heart is showing up. Why do you need the heart? To get going early.
Start pumping the blood?
You've got to pump that blood, right? Yeah, and where's the blood coming from? Where's the red blood cells coming from? The yolk sac, exactly. Isn't that crazy? Yeah. So we got all of these structures really early on, just 1 month in, that are establishing themselves so that they can take care of it and help develop all the other systems. So here's an image of the heart, 15 days, so that's half a month. Here's 3 weeks. Here's 1 month. Not completely well established. Here's birth. So this is where we are there, about there, heart-wise, development-wise. Another image of that 1 month old embryo. Look, we have a tail. Just like all other species with tails, we've got tails. Lots of things are developing. The whole point here is that this is just 1 month in. 1 month in, a little bit closer view. Got your somites establishing. These pharyngeal arches link us to the gills of organisms that live in the water. Yep. We got ears starting to develop, the eye, there's your forebrain or head, there's medulla oblongata back there, there's the tail. I don't know what happened to the little L there. But clearly much is happening in this first few months. Week 8, so 2-month-old embryo. Now we have an established amniotic cavity with the fluid, amniotic fluid. The membrane itself, remember, is called the amnion. The space is the cavity, and it contains amniotic fluid. Got an established umbilical cord. The placenta is getting established. It takes at least 3 months for it to be fully established, and we can let go of the chorionic or the corpus luteum entirely. Week 12, so this is 3 months. Little fingers, they have not been separated yet. They're webbed. Did you know that you have webbed toes and fingers? Some people are born with those webbed toes and fingers.
Phelps, wasn't it?
Oh,
One of the big swimmers
I don't know.
still has webbed feet, or slightly webbed feet.
Really? Yeah.
Which is
Weird.
Olympic.
My father apparently had webbed toes. I never saw his toes. Seriously. So when I was really young, he would go to the beach, but he quit going to the beach with us right when I was 5, 6. I mean, I never saw his feet. He wore shoes all the time. Probably because he was a little weird about his webbed toes. It was weird. Yeah. Pardon me? No, okay. So here's another lay of the land, just giving you a sense about days. I had to work it all out here. So in 37 days is 5.5 weeks. 42 days is 6 weeks. There's 6 weeks. 49 days is 7 weeks. And then we got 50, that's 8 weeks. This is 7.5 weeks. And so between 49, where we just have webbed fingers, now we start to see some separation of the fingers and the toes. Toes finally separated at 56 weeks. If you are really interested in the entire growth and development of the embryo through to the birth at 9 months, here's a great website for you. I actually haven't checked it out lately, but if it's not working, you'll let me know. I haven't looked at it lately. And so babies can be born on time, but babies can also be born a little too early. Babies like to hang back too, so when they're hanging back, you know you're going to have an induced pregnancy or an induced birth. But if they are born prematurely, there can be some problems with that premature birth, especially depending on how premature it is. So they would like to have a baby that weighs about 5.5 kilometers. Kilograms, 5.5 pounds.
Baby.
Kilograms would be way too much, 5.5 pounds. Things that can happen when they are born too early, like before 7 months, which, by the way, my sister was born at 7 months. And she was born when they didn't realize that oxygen tents killed eyes if you use pure oxygen. She was put in an incubator with pure oxygen, and sure enough, it destroyed her eyes. So she's always been visually deficit, always wearing glasses. But that's neither here nor there. So things can happen. One, respiratory distress. And respiratory distress happens because the lungs aren't fully developed. They're not ready to go. So they have to get those lungs ready to go. Because their lungs aren't fully developed, they're not producing surfactant. You remember what surfactant was about? Mm-hmm. And so if they don't have their surfactant in the lungs, then they can't prevent their lungs from collapsing and sticking. So we need that. And then also that surfactant allows for oxygen and CO2 to diffuse more readily. So what they do is they literally put surfactant down the trachea and into the lungs. They add synthetic surfactant, and that helps with that. Babies have a hard time thermoregulating. They can't manage their own temperature because their hypothalamus isn't ready to go, right? The hypothalamus is the one that says, hey, you're too hot, hey, you're too cold. You've got to do something about that. They have a difficult time with the digestive system, again, because it's not fully developed. And so they have a hard time suckling because they don't have the muscular strength in their mouth and the ability to swallow, difficult to swallow because, again, musculature. There was a time when they believed that breast milk was bad for prematures, and then you had to feed a special formula, but fortunately they've come around to realize that nothing is better than the original, which of course is breast milk.
Who came up
Because
with
it's
that?
easily digested. I don't know,
Formula
money,
companies.
money, money.
Formula companies.
Money, money, money. Yep. Babies' livers may not be ready to take on all of the jobs that the liver has to do. And so you might end up with a baby that looks a little yellow, a little jaundiced, and even full-term babies can end up being born a little jaundiced, which a little phototherapy helps by oxidizing the bilirubin and getting rid of that bilirubin through urination. Right? So the liver is having a hard time with those plasma proteins as well, and that processing of the bilirubin is why they turn a little jaundiced. Things that can lead to this premature, but originally the causes were all blaming the mother
Of course they were.
from this original slideshow, and I had to adjust that. So a short cervix. Maybe you can't hold on to that pregnancy as long because your cervix isn't strong enough, not muscular enough. Mother who smokes or drinks excessively, understood. Smoking and drinking, not good for pregnancy. Diabetes, can you blame that on the mother? I don't know. Diabetes mellitus, sort of depends what kind of lifestyle goes along with the smoking and drinking. Poor prenatal care, I don't know that you can continue to blame this on the mother. I think this has a whole lot to do with society.
Exactly.
Yeah. Drug abuse, maybe you could blame it on the mother, but also again,
Society.
it's societal. Young or old mother, you definitely can't blame this.
Old mother is about 35.
Well, I hate to tell you, but above 35,
It's considered
you
geriatric
know,
pregnancy.
it's the
so
ovaries and the ovaries and all
Yeah.
those old oocytes. Old oocytes.
It's not,
Are
it's just like.
potentially reasons
Seriously?
for premature birth. This I find fascinating, how we actually produce this baby through this narrow canal called the birth canal. It is called labor, and labor begins when the amniotic sac breaks and your water breaks. That's the first sign of labor. What is it that's stimulating that labor? The release of oxytocin. What is it that drives the release of oxytocin? Stretching of that smooth muscle. Yeah. So you get this oxytocin flush, and then this baby hopefully is in the correct position. We learned a week ago about the different positions that a baby can take, transverse, breech, etc. So hopefully the head is pointing down, and that baby is ready to come through the birth canal. The cervix has to dilate. Cervical dilation, they like to see at least 10 centimeters or greater. So what's 10 centimeters?
A lot.
Right? Not so much, really. Yeah. Yeah, about that, Ilse. Yeah. So we got the rupture of the sac, we got the dilation of the cervix, and then the head, this is the cool part, the head turns sideways. So it's not facing down as it would be in the uterus, but it turns sideways because the head is flatter that way.
Yeah.
So it can slide through the cervix a little more easily. So once the head turns and gets through the cervix and down the vaginal canal, then the shoulders get turned in the
Or they
same
don't.
way. Isn't that cool? Instead of this way, instead of coming out like that, they go this way so that they can manage that shape of the birth canal. Because the birth canal is oval this way. It's not round, and it's not oval this way. It's oval laterally. Remember this from looking at the pelvises. Yeah.
Or you don't, and you cause problems. Or you don't, and you cause problems.
Yeah. So the shoulders go, and then the rest of the body pretty much slips through. Once you move the broad shoulders through, the rest of the body slips through. And lastly, the placenta is released. That's called the afterbirth. The great YouTube video, haven't checked it out lately, you should, to see all the cardinal rules, all the cardinal movements of labor, which is what they're called, cardinal movements of labor.
Isn't there a thing where sometimes the placenta doesn't come out and you have to
Yeah.
go and get it?
Yep. Yeah, further expulsion. Yeah. All right, so once that baby is out, then you have it attached to the umbilical cord. What's the umbilical cord doing for that baby?
Nutrients.
Delivering nutrients.
And blood and oxygen.
Oxygen in the blood, for sure. What else?
Taking wastes.
Taking away waste. What kind of waste
Carbon dioxide.
Carbon dioxide. And so the cord is cut, which means that the levels of CO2 are going to increase. What's that going to do to that baby's respiratory system? It's going to drive that first respiration. Why? Because now you have CO2 building up. Remember, we're constantly producing CO2. We're constantly getting rid of it. Every expiration is getting rid of extra CO2. So we have CO2 levels that are going to drive those first breaths. It's hanging the feet upside, or hanging the baby from the feet and patting it on the bottom is not the drive for respiration. But what is that value of that? What's the value of holding it upside down?
I could not possibly imagine.
Did I hear mucus?
Yes.
Fluids, right? Those are fluids. The baby was living in a pool of fluid. You got to get rid of the fluid. So if you want to, you need to, if that baby's having difficulty, once the umbilical cord has been ligated and it's still having difficulty, you might want to pull feet up so that some fluids can drain out of that respiratory system, especially out the trachea. Cardiovascular system. Oh, so breathing rates, I think that's fascinating. 45 beats, 45 breaths per minute initially. That calms down, as you can see, after about the first 2 weeks. So really fast respiratory rate the first couple of weeks, which I think is valuable because you need that extra oxygen to maintain your ATP levels, because what is going on in that baby? You're talking
Growth.
mitotic division like crazy.
So much growth.
So much growth, so much growth. Cardiovascular system, so we remember the foraminal between the right atrium and the left atrium. That closes up immediately, becomes the fossa ovalis. Remember that you have the ductus arteriosus,
Ligamentous.
becomes the ductus ligamentus. Yeah, ductus ligamentus. And then the ductus arteriosus becomes, it'll come to me. There it is, ductus arteriosus. It becomes oh God. All right, the umbilical cord, that's the ductus ligamentus, becomes the ligamentous teres, or actually the falciform ligament. Do you remember the liver? You had the falciform ligament and the round ligament?
Yeah.
That round ligament is the umbilical cord's remnant, and the falciform is the remnant of that the umbilical, or ligamentum arteriosus. No, yeah. Oh, there it is, ligamentum arteriosus. All right. Pulse rate's super fast. That's all going to slow down along with that respiratory rate. Of course, big production of red blood cells because you need oxygen. You got to get that oxygen to the tissues. What other things can happen? Takes a while for this baby to get transitioned. I'm sure those of you who have babies or have had babies know about these things. That transition period, making sure that the heart rate, the respiratory rate slow down after several weeks. Maybe some building up of mucus and debris, gagging, breathing problems. What do you do with them? Same thing. Pat, 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 make sure that everything is open and able to drain. Immunology, in terms of how they are going to be able to deal with the exposure to all these new proteins, these new foreign proteins, while they were in the womb, while they were in the uterus and the placenta, they were receiving the IgG, the immunoglobulin G, which is being produced in the mother, travels through the placenta. Do you remember how that happens? That transplacental movement? So the protein is encapsulated, right? It's endocytosed into the placenta and then delivered to the fetal blood by exocytosis, carried through the placenta. I'll have to show you, remind me of that one. What else? Oh, and then breast milk is going to deliver some immunoglobulin A, and that, of course, is going to carry on until that immune system gets up and running on its very own. And you who have children are experiencing immune systems that haven't been exposed to certain foreign proteins, yes? Do you have children that are coming home sick? Anybody with children? Sick children?
I was a sick child.
You were a sick child. Anybody else? No? No other babies in here?
They're all babies in here.
They're all babies. Good call, Clarissa. What other things can happen? So congenital anomalies. What kinds of organisms can cross through the placenta and get to the fetus?
HIV.
Herpes. Herpes simplex. HIV. Rubella, German measles, one of those that you can prevent now with a vaccination. Cytomegalovirus, I didn't know anything about cytomegalovirus, but apparently 50% of all of us have been exposed to and infected with cytomegalovirus. It is very common and it's part of the herpes family. And it's not necessarily a horrible virus. You know, it doesn't kill people. It just makes you a little sick for a little while, maybe, or not even. Maybe you've been exposed to it and you don't even respond to it.
Listeria is a big one too.
Most show no signs or symptoms. Yeah. So cytomegalovirus, very interesting. Toxoplasmosis, have you heard about toxoplasmosis? What do you know about it?
Cats.
Cats.
Yeah.
Infected feces.
Yes.
And
Pregnant women should not
do not scoop out kitty litter, but you can find it in your garden as well.
Oh, yeah.
Anywhere that cats like to poop.
Yeah. But you can also find it on your meat cutting boards.
Ew.
Oh, yeah. Oh, yeah.
What is it?
Yeah, because yeah. Well, so toxoplasmosis, for those of you who don't know, is a blood parasite. And cats are the definitive host. I mean, they're the ones that are carrying the toxoplasma organism that is going to then produce life stages, let's say. So the life stages are carried through circulatory system and then through the feces, and they're delivered through the feces into the soil or the litter box. And it used to be that if you were pregnant, your pediatrician would tell you to get rid of your cat because you were so at risk of getting toxoplasmosis, which is bunk.
Extreme.
a bit over.
Extreme bunk. Yeah. Because you don't have to clean the litter box. And if you're gardening and you have a cat and your cat defecates in the garden, you wear gloves while you're gardening. And while you're not cleaning the litter box, have someone else do it. If you are cleaning the litter box, you clean it daily. Because it's the dust that carries those little, they're not oocytes, they're cysts of the life cycle that you can inhale and then develop toxoplasmosis. So don't worry about toxoplasmosis. It certainly can be delivered, from you to your fetus, but it's not something that you need to get rid of your cat for. Just FYI. Other things to worry about, and these I think are far more worrisome, right? Teratogens, chemicals, other things that you can't control in your environment. All the chemicals that we're exposed to. Gotta be careful about that. There was a time when thalidomide was considered the drug for women who were suffering from morning sickness. Unfortunately, thalidomide also resulted in fetuses that were born with unformed limbs, arms and legs. And curiously enough, thalidomide has made a new resurgence in that it is actually had been identified as a good cancer-fighting agent. I think I've written down here which cancer it is that they're using it for. Yeah, multiple myeloma. Yep. Sedatives, barbiturates, opiates, all those things are things that you could live without while you're pregnant, I think. That goes back to blaming the woman for, premature birth. Accutane, does anybody know what Accutane is?
Yeah,
Have you used it?
yeah, and literally on the pill, on every single pill, it has "do not take while pregnant," like every single one.
Yeah, because it can do damage. And then, of course, we know about fetal alcohol syndrome. That's not to say that the occasional 2 ounces of a sip of wine is probably not going to cause fetal alcohol syndrome to your fetus. But of course, if you're drinking a bottle a night, maybe you should be. So moderation, it's that kind of thing. Smoking, x-rays, all those things can cause problems in the development of your central nervous system in particular. And so I bring this to your attention because it shows you how far and what is happening with your central nervous system, right? So all the impacts that can occur while you are pregnant is especially important for your central nervous system development. Heart, well established early on, central nervous system, well into the 4th month of pregnancy, into the 2nd trimester. Just as a way for you to appreciate the kinds of things that are developing and at what time frame they're developing. I think it's especially important to note the central nervous system. So we talked about the 3 trimesters. We have trimester 1, first 3 months, 2 and 3. The first 3 months are really the most critical because that's when all your organ systems are getting established. The 2nd trimester, 3rd trimester is really pretty much just increasing size, gaining weight. Things can happen, of course, that you have absolutely no control about. That includes mutations or genetic anomalies can be resulted from anything extraneous, right? Epigenetics is a biggie. You can be exposed to foreign proteins, you can have all kinds of things that can change the genetic makeup, causing mutations. You can have non-disjunction of chromosomes. That's the next thing we're going to talk about. You can have non-disjunction of chromosomes of your sex chromosomes, and you can have non-disjunction of chromosomes in your autosomes as well. And one of the most common of those is trisomy 21, which we learned is called Down syndrome. And I learned this weekend that Down syndrome is named after a man named Down. His last name was, I think his first name was Samuel Down. And the reason Down syndrome was named after him was because prior to his developing an interest in children with Down syndrome, children with Down syndrome were institutionalized. And they were stuffed in the most horrific conditions. Right? Too many in a room, beaten, abused, starved, horrible conditions. And he saw that, recognized that. He went to one of the major institutions and took over as the president of the institution and changed everything around. He increased their nutrition. He changed the way they lived. He, instead of beatings, he praised, and he introduced children to art and music and animals and all of the things that make life worth living. And after he left that institution, he opened his own, and it was sort of like this place called Green Chimneys, where children with learning disabilities are engaged and lifted, and he did that in his own place. And because he made such a significant difference in the lives of children born with trisomy 21, they renamed the condition after him, and it was, I think, 1964 and 1967. I mean, recent history, when they renamed it and called it Down syndrome because of the impact he had on recognizing these children and recognizing their abilities rather than their disabilities. Anyway, so we can have Down syndrome, that's called a non-disjunction of the autosomes, and we have so many things that can happen with non-disjunction of the chromosome, or the sex chromosomes, which is really fascinating. So this is your normal, what they call disjunction of the X chromosomes. Remember, your oocyte carries X chromosome always, always, always. The sperm carries an X or a Y. And so in this case, the parental cell is a female cell, and this is meiosis happening here. In which case, when meiosis happens, the two X chromosomes get segregated, and each of those X chromosomes can be fertilized either by an X sperm or a Y sperm. If it's an X sperm, you get XX, normal female. If it is XY, you get XY, normal male. This is pure biology, right? Pure biology. I could say masculinized, feminized, but I don't think you guys care about it. Good. What happens if we don't get that separation? What happens if in that first meiotic division we don't have the X's separated? And this oocyte gets two of them, and that oocyte gets none. This is fertilized, you get an XXX, a triple X. This, by the way, is amazingly common, like 1 in 5,000. Crazy. If you don't have an X in there already, but there's an X delivered, it's XO. Oh, this, I'm sorry, Turner syndrome, this is the one that's really common. XO, really common, like 1 in 5,000. Triple X not, I don't think that's quite as common. Here's all the kinds of things, and there's more. This is just, right, there's more. But this is kind of the more, what we're well familiar with, non-disjunction of our sex chromosomes. So we know about trisomy X now, infertile female, right? Klinefelter syndrome, where the double X is fertilized by a Y. Sperm fertilizes the double X. In Klinefelter syndrome, you have a male looks perfectly normal, but he's sterile, average intelligence, as it says, underdeveloped testes can lead to the sterility. Klinefelter syndrome. Turner syndrome, where there is no X in the oocyte, fertilized with an X sperm, get X0, no X in the chromosomes, fertilized by X sperm. This produces a sterile female, but without any secondary sex characteristics. So no breasts, right? No broad hips, the kinds of things that you would use to identify a female. 1 in 2,000 female births. Is that crazy? 1 in 2,000. I think that's amazing. There's a foundation for it. That's how common it is. The Turner Foundation.
What's a webbed net?
Yeah, what is that?
It's widened. Looks like that, if you can imagine.
You can kind of see the skin stretching from the neck down
Yeah.
to the shoulder.
Yep. Yep. The tendons, the musculature widened. No breasts, no secondary sex characteristics. Jacob syndrome, this is the XYY that we were talking about earlier. And Jacob syndrome is going to produce a normal, generally looking individual, normal fertility. As you see here, maybe some learning disabilities, maybe an increase in acne. Other things that I learned about it is that you have these individuals, these gentlemen with Jacob syndrome, they can produce children that are completely normal, with normal chromosomal distribution. Turner and Klinefelter are some of the most common genetic conditions in humans. So this term aneuploidy, why do I want to hang on to it? It means that there's an abnormal distribution of chromosomes. Either they're left out or they're increased in number. So abnormal number and distribution of chromosomes. So that was the non-disjunction of the sex chromosomes. These are some of the non-disjunction conditions of the autosomes. I don't know if any of you have ever been to this place in Portland called the Providence Medical Center for Fragile Children, I think it's
The
called.
Child Center.
Child Center. Have you been there?
I work there, yeah.
You do?
I work at Providence Portland.
So you're familiar with that particular region? Yeah, isn't it amazing?
Yeah, it's most of the time, I don't usually go and see like actual children. It's mainly a place for adults who are kind of like, they don't have family to get to help take care of them. And
Yeah.
so after discharge from the hospital, they go over there.
Yeah,
But they don't have a place to live
yeah,
or anybody to take care of them.
yeah, and you wonder like what happened.
Yeah, we do get it sometimes, cause I work over in, the X-ray department, and so we sometimes overdose tubes for children, tubes for adults.
Yeah. It's humbling.
It is,
It's humbling, and if you are at all interested in working with children with disabilities or adults with disabilities, this is an important place for you to go and visit and potentially volunteer,
100%.
because they need a lot of help.
Yeah.
They are really, they're disabled,
Mm-hmm.
Yeah. and they have a wide range of problems. Most of them they're profound.
Yes.
They're so profound, they need to stay in a facility where you have medical help and the equipment and the tools that are needed to take care of these people. But the two that we're gonna talk about, aside from Down syndrome, who we now know is named after Mr. Down, trisomy 21, meaning that you have 3 chromosomes, 3 of the 21 chromosomes, number 21. Edwards syndrome, AUC, is trisomy 18, very short lifespan for these guys. And then Patois syndrome for trisomy 13, which is pretty much lethal, pretty much right after birth, 7 to 10 days, as you can see. There are other things that can happen to these chromosomes as well. Maybe you remember from your Y112 class how you can have mutations of those base pairs, where you may be getting flipped, or you have a base pair is missing, which changes the sequence of all your base pairs down the line, or you have an insertion, an additional base pair that's tucked in there, again, changes the sequence down the line. Remember that our sickle cell anemia is the change of one base of a base pair, which impacts the production of hemoglobin, which of course impacts the ability to get oxygen to your cells. So all these kinds of things can happen. You just never know what's going to happen. Like I said, here we are. We're sitting here. Who knows what little things are going on with us? But basically, we're all able to walk, talk, eat, drink, pee, poop, function. I think. Am I looking at everything? Pretty much the same. Yeah. So Down syndrome has some characteristics that are classic. One of them is this fold on the medial canthus. Another is the curve of the pinky finger. Another is the short hand, but also the single palmar crease. Let's look at our crease. How many creases do you have? I have 2. We have 2. 1, 2, 1, 2. Yeah. So if you don't have them, maybe you have Down syndrome. And by the way, you can have like a Down syndrome that is really mild. There's 3 different ways that Down syndrome can be impactful in a body. So those people who have this medial canthal fold, maybe an enlarged tongue, might have noticed. Does anybody know somebody with Down syndrome?
My sister. I have a sister.
You have a sister with Down syndrome? Is she profound, or is she?
I mean, she can't take care of herself.
Uh-huh.
I mean, she's like a 10-year-old, I would say, with like a 6-year-old reading level and maturity level.
Oh, so she has been pretty well impacted.
Yeah, I mean, we all have to take care of her. She stays with my brother.
Uh-huh.
And she stayed, yeah.
Oh.
I mean, she can't live by herself.
No. How old is she?
Right now she's 50.
Ah,
Yeah,
yeah.
she's my older.
But sweet, I'm sure.
Super sweet,
Super sweet.
very stubborn. All she wants to do is listen to music and dance and talk about famous people.
Okay.
Yeah.
There are worse things in life.
Yeah. No, that's what she loves. She loves movies and famous people.
Happy.
Very happy.
Right?
Yeah.
But if you leave
What
her
a relief
alone,
if all you have.
Yeah.
If you leave her alone,
that's the
she'll
only thing
eat.
you're worried about.
she'll eat everything in the refrigerator. She'll eat like ketchup sandwiches. She'll just we've had to like lock the refrigerator for her because she does not have
Boundaries.
impulse control.
Right? Interesting. Well, so you all understand this is where your trisomy 21 comes into play. And then now we're going to talk about inheritance. How does that all happen? Well, hereditary traits. Did anybody go and look at genetics? Patty, do you learn genetics? Did you like it? Wasn't it cool?
I love genetics.
So many cool things in there, right? It's a great
So
website. So you all know that we have 23 pairs of chromosomes. We have chromosomes from our mom, 23 of them, 23 from our father. And if you're a male, you got the Y chromosome from your father. If you're a female, you got the X chromosome from your father. we have 23 pairs of these chromosomes. These pairs are called homologous chromosomes. Homies, I like to think of them as called homies. Homie 1 matches homie 1, right? Homologous chromosomes. And on those chromosomes, we have traits carried via the genes. And the traits on one chromosome are referred to as the allele of that trait. Don't forget that. The allele is where that trait is on that particular chromosome. The allele for that trait on the other chromosome may be the same. In other words, you have two dominant alleles for a trait. You have homozygous dominant situation. Maybe one of those alleles is different, and it's the recessive expression of that trait, in which case you have one dominant and one recessive. You have a heterozygous situation. If you have two recessive alleles, then you have homozygous recessive. And remember me telling you that being recessive doesn't necessarily mean it's bad. Being dominant doesn't mean it's better. It's just dominant versus recessive, because 5 digits on the hands is a recessive trait. There are many traits. I think like around 200 of the traits are what I consider Mendelian traits, or complete dominance. The dominant gene overrides the expression of the recessive gene. Then there are other conditions whereby these traits get combined with themselves. So we're going to look at some of those. We all remember the terms genotype and phenotype, right? Genotype is the genetic makeup, phenotype is how it's expressed, what we see. And we played with our Punnett squares. This particular Punnett square is showing you what happens with somebody, with two people that are carrying the trait for PKU, which is known as phenylketonuria. Phenylketonuria is the inability to process phenylalanine. And phenylalanine, if it builds up, causes some major problems. This is one of the things that your kidneys get rid of, right? Can't metabolize and get rid of these phenylalanine proteins. Let's say you have two humans, two people that are carriers for this particular trait. And as we worked in lab last week, you notice that we can have a 25% chance that these two people crossing can produce a child with phenylketonuria. That's a problem for that individual. But that's not saying that they're going to always produce a child with phenylketonuria, or that they will. This is simply saying they have a chance of doing that. They have a 75% chance of producing a normal individual, and a 50% chance of producing a carrier. Remember, a carrier is carrying that trait that isn't necessarily being expressed. And then we've already talked about this non-disjunction problem. Non-disjunction of our sex chromosomes, one problem. Non-disjunction of our autosomes, yet again, additional problems. We can have problems with something called translocation, and this is actually one of the ways that Down syndrome can be expressed, is through translocation, where a chunk of one chromosome attaches itself to another chromosome. And that happens in trisomy 21, right? 3 copies of that part of chromosome 21 is attached to another chromosome. And we can have lots of mismatches that don't get fixed because your enzyme that goes along and replaces
Polymerase.
the...
DNA polymerase.
Yeah, DNA polymerase, that's right. They are capable of going along the length of your DNA and making sure that all your matches are lined up correctly. But sometimes they get through and you end up with mutations. So that's, your complete dominant situation, things can happen there. This is a nice way of understanding incomplete dominance. We talked about incomplete dominance relative to sickle cell anemia. People with sickle cell trait have incomplete dominance of that trait. In other words, 2 people with the trait produced 75% chance of producing somebody normal or appearing normal, and 25% chance of producing somebody with the condition. But these 2 carriers, they can be challenged. if we have a sickle cell anemia, an individual that is a carrier, it could be fine at sea level, but when you take them out of sea level, where there's plenty of oxygen in the air, and you take them to a higher elevation, they might have difficulty because there's not enough oxygen to actually infuse their red blood cells. So carnations express incomplete dominance. You cross this red one with the white one, you get pink. Curly hair, wavy hair, straight hair, another form of incomplete dominance, kind of fascinating. The point is, however, that this is what's referred to as a continuous trait, so we don't have just curly hair, just straight hair, and just wavy hair. We can have any range in between. I have one of those couple of bends. Yeah. This is just the expression of that sickle cell anemia as we discussed in lab, and we just discovered again. So do you all understand how this is working? These sickle cells might look totally fine, but when they are exposed to low levels of oxygen, they turn into sickle cells because the hemoglobin isn't made correctly. They might pump back up again, but usually they don't. They stay sickled. We have codominance, and codominance in us is expressed by our blood types. So you can have type A blood, you can have type B blood, you can have AB blood, codominance, A and B. Or you can have O blood.
Not BO.
Not BO. No BO.
Because O is the lack of it, right?
O is the lack
lack
of
of-
proteins on the surface.
Yeah. So obviously, if B it would just be B, not B and O.
That's right, B, yes. A, B, B, or A. You can have multi-allele inheritance. So crossing an A person with an O person, multiple possible combinations. You can have polygenetic inheritance. We talked about this, I believe, that we have certain traits that are dictated by more than one gene. Polygenics, many genes for a particular trait. This slide came to us saying that there are 3 genes that drive skin color, but in my research, it looks like there are maybe 5 or maybe 100 different genes that contribute to your skin color. Can you imagine trying to work out a Punnett square for just 3 genes? This is the Punnett square for just 3 genes. How did we get from big A, big B, big C, to big A, little b, big A, little a, big A, big B? How did we get there? Meiosis. Crossover. Independent assortment. Yeah. These are of course, the theoretical. These are theoretical crosses. Pleiotropy, fascinating one. This is where you have one gene that impacts multiple traits. Albinism is a result of pleiotropy. And albinism is not so much identified by your skin color and your hair color and the lack of melanin, but more about the eyes, believe it or not. So people with albinism have difficulty with their eyes. Vision is a big problem for them. The retina isn't formed correctly, and they may have crossed eyes, or they may have eyes that drift to one side or another side, or lazy, as it's called, lazy eyes. Yeah. There's another syndrome called Marfan syndrome, that the gene that dictates the production of fibrillin. Of course, fibrillin of the fibrinogen, fibrin family, connective tissue. You know that we have connective tissue everywhere. It's like the most common of the proteins, especially collagen. By the way, this is considered autosomal dominant, just like Huntington's disease is autosomal dominant, meaning that 50% chance you're going to produce an individual with Marfan syndrome. If you have Marfan syndrome, you're going to carry it, it's going to be carried on. Marfan syndrome results in lots of disparity in development. Tall, slender, really disproportionate length of arms, fingers, and legs. Turkey breast, where the sternum is protruding or dips inward. You can have problems with your palate and the size and shape of your mouth. A heart can be impacted, so murmurs are the result of valves that don't close correctly. Yeah.
They test every NBA player for Marfan syndrome.
Do they?
Because I think the the normal, I think what I read was the, like, there's like a basic, like height ratio
Uh-huh.
that they use in almost every NBA player qualifies because it's long arms.
Oh, right, oh, that's interesting.
They, like, genetically test everybody.
Huh, so that guy, the Asian guy, what's his name, the tall, super tall guy? Yami. Yamin, yeah. So he must have been tested.
They
Because he was-
they test everybody.
They test everybody.
Like, I have a friend that's a doctor, whenever we watch NBA guys, like, that guy has heart, like, I'm telling him that they test people, but he doesn't believe it. He
That's
thinks
it.
they all have heart.
He's a doctor,
Yeah.
interesting.
Wait, they're not allowed to have marker play? Why do they test them?
Because they have like heart issues. So there
Oh.
was one guy that was gonna be a draft pick and they disqualified him.
Uh-huh.
Because he has heart issues.
Because he actually has Marfan syndrome. So
Oh, that's so
they wouldn't clear him to
sad.
play.
Huh.
That's too bad.
Like
They
that.
don't want him to have a heart attack on the
Yeah, like-
You can't blame him for that, yeah.
Women with Marfan syndrome, most likely you can't, have a child, like, you can't give birth. Cause there's like a very solid chance you'll die during birth.
Uh-huh.
Because of the heart issue.
Because of all the there's connective
So,
tissues everywhere.
If one of my best friends, she has Marfan syndrome,
Uh-huh.
and if she ever wants to get pregnant and have kids, she needs to have open heart surgery.
Wow.
That's actually crazy.
And so it's like if she doesn't accidentally
Fertilization
get
there.
pregnant, like she has to,
Mm. Wow, interesting stuff. Yeah, Marfan syndrome, it's a biggie apparently. I didn't realize that there was-
Like normal, and
I
like,
didn't
getting into
look for the
NBA,
frequency of Marfan
And they
syndrome.
test you for something,
It
and
was
you
like
just lived your whole life
7 years ago.
and now
They disqualified.
And, like,
him. So they
dedicating,
said,
like, your whole
they did all the reports. They're like,
Yeah.
we do auto testing. for
Oh,
all
testing now for that, huh?
So is this picture of somebody with albino and then the
Well, this is another genetic combination
I
called
think he has
epistasis.
albinism too because of his eyes.
Epistasis is where one gene overrides the expression of another gene. My favorite is to look at Labrador retrievers. Because I love them. I have 2 of them. And so the black is dominant, big B. The chocolate is recessive. The yellow is the yellow because of a gene that overrides the expression of the color gene. It's called the epistatic gene. So this black lab is big, 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 big. It could be big B, little b too, but he's big E, little e, or big E, big E. This chocolate is little b because this is recessive, homozygous recessive, and this dog could be big E, little e, or big E, big E. This lab with the black nose and the black mascara is big B, or big B, little b, but he's got the homozygous recessive epistatic gene that overrides the expression of the color gene. Same here. A little pinker on the nose, maybe you know these guys? A little pinkish on the nose. Still the black, but still a little, so big, little b, so even this is less color. And there is your epistatic, homozygous recessive epistatic gene, overriding the expression of the color gene. It happens with individuals who have the balding gene. And then the balding gene, this balding pattern is X-linked, and it is about the dihydrotestosterone levels for the gene that produces dihydrotestosterone, and they have low testosterone levels, but average dihydrotestosterone levels, and those with the same amount of dihydrotestosterone as others, but their follicles are genetically programmed to be sensitive to dihydrotestosterone, and they drop their hair because the follicles are responding to that gene that's controlling the follicle and the growth of the hair.
Bless you.
Yeah, isn't it? Interesting. Epistasis. These, by the way, are not part of your learning objectives in the lab. This is just because I find them interesting and they're life applying, things that you can associate with. So autosomes and sex chromosomes, we all know about this. We know females are XX, males are XY. Y is a smaller chromosome, but that's okay, it's still needed, and they are not going away. In case you've heard anything about the Y chromosome eventually disappearing, it's not going away. It's going to stay, but it has a limited number of chromosomes. It's very true. Because its primary job is to deliver that sex-determining region to the developing embryo. We know that we have 23 pairs of chromosomes, 22 of them are autosomes, one pair of sex chromosomes. What do we call these pairs? Homologous chromosomes, right? They're homologs. Homies. Can't use that in the exam. If there is a question about them in the exam, you can't use homies. When you're talking to others, you can use them and they'll be like, oh, that's cool. So we have, of course, sex-linked inheritance that happens within our bodies. We're familiar with the fact that many of them are genes that are found on the X chromosome, and then we can blame the X chromosome. And the X chromosome carries the gene for color deficiency. We don't call it color blindness anymore, it's color deficiency. Also, hemophilia is a sex-linked, X-linked condition. Right? We know what hemophilia is all about now. Others that I found fascinating, the absence of incisors is excellent. Who knew that that was even a genetic condition, right? Night blindness, juvenile glaucoma, some kinds of deafness, nephrogenic diabetes. What do you think nephrogenic diabetes is? Something's wrong with the nephron, right?
I would assume they don't uptake insulin or glucose or something.
Something's happening in the nephron that they are not capable of managing their glucose, right? Cataracts, genetic, muscular dystrophy, and male pattern baldness are other sex-linked traits. And you're familiar with all of this now. And everybody feel good with this ability to work out these Punnett squares? Okay. I remember me telling you how females have two X chromosomes. Well, we don't need two X chromosomes in all our cells, and so we inactivate one of them pretty early on in the embryonic development. And once they're inactivated, they don't go away. They're just hung on to the they're within the nucleus, and just at this periphery of the nucleus. And you see here on this picture is a cell stained. Here's the bar body for that chromosome, for that cell. Here's a neutrophil, and I told you it looks like a little drumstick. So if any of you ever do any hematology, like you end up getting really into blood work, you can tell the person that you're looking at the blood whether they're male or female just by finding the bar bodies on the neutrophils. Because it's X-linked inactivation. What do I do with my thing? There it is. Other things that can happen to us. Environmental influences can make a big difference. We talked earlier about teratogens. What are teratogens? Anything that causes a mutation, a developmental problem, a defect. Chemicals, drugs, cigarette smoking, radiation, radioisotopes, anything that can influence, especially during that first trimester. Remember how far the central nervous system goes in terms of development. Anything that can impact that can cause problems with development overall. And here's a little more about Down syndrome. And the point about Down syndrome that I wanted to bring up is that we have three kinds of Down syndrome. There's non-disjunction, which literally one X chromosome ends up in the other cell. So you end up with 3 in one and 0 in the other cell. You have translocation, where literally a piece of chromosome 21 relocates and finds itself on another chromosome. And then you can have the mosaic, and this is the most fascinating, where you can have some cells with triple trisomy, but some cells completely normal, which is very interesting because you can have an individual that doesn't necessarily display Down syndrome with the mosaic. The Fragile X syndrome. So I haven't looked at this, I haven't reviewed this one, but it is interesting that we have normal X chromosome, or a chromosome called the Fragile X chromosome, which is the result of an increased number of base pairs, expansion, as it says here, of the CGG triplet, right? You know, you have the codons. This is the triplet, this is a codon. If you have an increased repeating of that CGG greater than 200, then you can develop this Fragile X syndrome, whereby you have this site which makes this whole chromosome fragile. Here's the things that can happen with it. Learning disabilities, double jointedness. Well, we know there's no such thing as double jointedness, but really lax joints. Yeah, so your ligaments and tendons really give way. Maybe more autism result of this. Not your Tylenol a pregnant woman. Mental retardation. And so you can see that this can pass from males to daughters, and from daughters to children, because the daughters are carriers of it, because the X chromosome is delivered via the male. Broken tip, genetic stutter. Increasing that base pair changes the way that chromosome is held together. All right, this is where we're going to stop. We'll finish this up. This little